Imagine for a moment that you are in the shoes of Antoine van Leeuwenhoek, the inventor of the microscope. And imagine that you took out your microscope and you looked at a plant leaf and you saw some cells. And then you looked at a slice of bone and you saw some cells. And then you looked at some yeast that were, growing on a petri dish and you also saw some cells. You might come to the idea that everything that was alive was made of cells. And this would be the very first part of a theory. What you've done is you've looked around, you've looked in all sorts of different contexts, and you've found some sort of pattern in the natural world. However, and when you said that, you actually would have articulated the first part of one of the most famous theories in all of biology, the cell theory, which is that all things are made of cells. But it's only the first half. Because in addition to describing a pattern that you've seen in the natural world, a theory also describes or proposes some kind of mechanism. How did that pattern come to be? And it wasn't until rather later that this part of cell theory came to gain some traction, which is that all cells come from other cells. So all of the cells in your body came from a single cell, a single zygote, which itself came from the merging and the fertilization of an egg cell by a sperm cell. And so I want you to compare this idea of a theory which talks about a pattern in the real world, which talks about a mechanism that might explain that pattern, to a hypothesis. And a hypothesis also has two components. First, it's an explanation of an observation, but importantly, a hypothesis is testable. And so let's compare cell theory, this idea that all things that are alive are made of cells and those cells come from other cells, to an observation, which is that if I leave some broth out on the tabletop overnight, things start to grow in it. Why is that the case? Well, I can use this theory that everything that is alive is made of cells, and those cells all came from somewhere, to develop a testable hypothesis, to develop a testable explanation of that observation. And so I might develop the hypothesis that those cells came from, they just were floating around in the air and they fell into my broth. And then they were happy and they had nutrients and they started to grow. And so my hypothesis for why my broth grows things is that those things came in from the air. And now, once I've developed this hypothesis, I can develop an experiment that tests it. And so I might imagine taking some freshly made broth and putting a cap on it and boiling it for an extended period of time and setting it right next to my open freshly made broth and asking, okay, the closed container isn't having anything fall into it. Does that prevent things from growing? And that prediction is important, right? I predict that if my hypothesis is true, then I will see something in particular. That good hypotheses are not only testable, but they actually will suggest what the outcome of an experiment to test it will be before you even do that experiment. And this is really key, because when we think about how biologists work, how they think, how they study biology, biologists use evidence. They use evidence to make decisions. We ask questions about how organisms work. we pose hypotheses to answer those questions, and then we use experiments or observations to collect data that support or refute that hypothesis. And if you do this in many different contexts, and you see many different observations that are all traced back to the same mechanism that explains them, then you have a pattern and a mechanism which can be wrapped up in a theory. And so now that we've talked about one of the most famous theories in all of biology, we're going to turn our attention to the other one, which is the theory of evolution by natural selection.